Good morning, class 9th. Today's topic is cell division. As you can read on your screens, it is written as cell division. That means today we are going to study about division of cell. That is how a cell divides. Are all the cell divisions similar? Are new cells produced by division identical to their parents, or they are genetically different? So many questions arises. But before answering these questions, let me explain the structure of chromosome once again. Yes, now you can see the diagram of chromosome on your screen. Here you can see that there are two chromosomes together and they are totally same in size and shape, right? But they are different in color. Why? Actually, they are homologous chromosomes, as you can see here. What do you understand by homologous chromosome? Actually, this homo means similar or identical, and logos means loci or locus, that you study in maths as well. That means location they are talking about. So they are homologous chromosomes which are totally same in shape, size, plus location of gene. What is gene or genes? Actually, gene is defined as a structural and functional unit of DNA. That means a long thread of DNA consists of small or larger unit of gene, and each and every gene codes for some function, whether it is smaller in size or larger in size, but it codes for some function. So homologous chromosome means the similar and the identical chromosomes which have similar gene location. Okay, why they are present in pair? Because, and they are different in color too, because they are obtained from different sources. One is mother and other source is father. That is why they are called homologous chromosomes. Now you can see it is marked centromere. This location, this one and this one is centromere, and you can see these yellow color structures. These yellow color structures are called kinetochores. You have seen the plugs at your home, right? We plug them into sockets so that your appliance starts working as soon as we plug it, right? In a similar manner, these kinetochore work as socket. In these socket, your spindle fiber, very important structure for cell division, attaches and pull these chromosomes at opposite poles so that division will easily take place. Okay, synaptonymal complex is not important at your class level, but let me tell you once again. This is the junction, this is the joining point between these two chromosomes. Okay, and what do you understand by sister chromatids? Sister chromatids are the arms. These structures are called arms of a same chromosome are called sister chromatids because they are similar. That is why they are called sister chromatids. Here you can see some structure actually on the basis of position of centromere, that is center of we classify them into four categories. One is metacentric. It is not compulsory that the centromere is at exact center. It is present at nearby center. When it is present at nearby center, the condition is called metacentric. Okay, and when it is quite away from the center, the condition is called submetacentric. You can see these arms are almost similar in size, and here these arms are different in size. This one is larger one and this one is shorter one, right? Similarly, if I'll talk about acrocentric or subtelocentric, that means telo means end or corner. edge, right? So when the centromere is near the end, okay, and it's very far away from the center median point, then it is called subtelocentric. Here you can see very short arm and quite large arm. So this is the reason it is called acrocentric or subtelocentric. And in the telocentric, the center is at the end only, at the corner only. Here you can see there is no arm. Yes, here you can see there is no arm. So only one side of the chromosome having arm. These kind of chromosomes are called telocentric. This diagram contains the same information, but it is the new one for you. This is your metacentric chromosome. It is exactly the same, but here you can see some new structure. It is nothing. It is just the repetition of some of the sequences present in the sister chromatids. As they are copied, they look like bulb-like structure at the end of the sister chromatids and called secondary constriction. And this chromosome, because of this extra sequences of the gene or extra structure present over this head, or you can say sister chromatids, they are called satellite chromosome. Now our main topic is cell division. It is last topic of your chapter. Cell division is the process by which new cells are made. New cell have its own significance. If I'll talk about unicellular organism, these new cells are called daughter cells because the body of unicellular organism consists of only one cell, or you can say one cell represents its whole body. So if that cell divides, it will divide into two. Both are called daughter cells. So it have different significance in the case of unicellular organism. But if I'll talk about multicellular organism, so here the significance of this cell division is growth, repair of dead, damaged cells or tissues, and formation of some new things, new structures, plus formation of gametes. Now what is gametes? Gametes are spatial cells only limited to reproductive organs of the body, whether male or female. So gamete production is also related to cell division, and for that we will study a new way of cell division. Here two types of cell divisions are mitosis and meiosis. Very first cell division is mitosis, the process of cell division by which most of the cell divides for growth is called mitosis. If I'll talk about our body, you can see our hands, right, leg, or you have a covering called skin over there. If that skin get any scratch or cut, new skin covers it, right? The division is called mitosis, and thus we can say that it helps in growth. As we know, 5 years ago, we are smaller in size or height, you can say, but with time your height increases, right? So for the growth, as we know, we have billions of cells in our body. If our height increases, it needs more cells so that it can show the growth. So that growth is also done by this mitotic division or mitosis, we can say. In this process, each cell called mother cell divides to form two identical daughter cells. Two identical means they are exactly copy, you can say photostat of each other. There is no difference between them. Even if they have any disease, that disease will be copied as it is. So this division is also called equal division, equational division, because the number of chromosome is equal. Okay, so the daughter cells have same number of chromosome as mother cell. It helps in growth and repair of tissues in organism. This diagram is also present in your book, but before explaining this diagram, see one thing. Diagram number 1 and diagram number 5. Though the diagram number 1 is single and 5 have 2, both are similar. They are exactly similar. This is the mother cell and this is the daughter cell. These two are daughter cell, right? So here in next steps, that is step 2, the chromosomes are doubling themselves. Similarly, if I have something with me that is sufficient for me, but in case if I have to divide it in 2 equal halves, if that thing is sufficient for me, how can I divide it? So before dividing it, I'll double that material so that I'll get the same amount which is sufficient for me, right? So here also the chromosomes will double the material, then they will, after doubling the material, here you can see the dotted lines. These are called spindle fibers. These they align along the spindle fibers through that kinetochore which I have shown you in previous diagram, and through that kinetochore these spindle fiber pull their part of chromosome towards them, that is towards opposite pole. This is pole number 1 and this is pole number 2. So both are opposite to each other, and that is why they are called opposite poles. So these spindle fiber pull the chromosome towards opposite pole. Once the chromosome reaches their pole, the cell divide. Okay, here you can see the constriction. This constriction I'm talking about, it will increase in size slowly. It will come near, and once it come near, the cell will fully detach from each other and become 2 daughter cells. But in the case of plant cell, this cell division or you can say the division of cytoplasm takes place by cell plate formation. Cell plate formation means, for an example, if you have a large room and you want to divide it into 2 equal rooms, okay, in that condition what will you do? You can't make wall closer so that it will divide into 2, as in the case shown here, right? This is the case of animal cell. In this case, what will happen? We will make a wall. We'll start attaching or joining bricks together, and finally it becomes a wall. Divide the room into 2 equal rooms. The same case can be seen in plant cells. Their cytoplasm divides by cell plate formation. A plate-like structure forms between the cell and it divides both the cell into 2 equal halves. Okay, all other steps are same, only the division of cytoplasm, also called cytokinesis, is different. Here is some informative diagram for the mitosis. It is also called somatic cell division, and another name is equational division because it is equal. Okay, so kindly go through this diagram. If you want to ask any of the terminology, please feel free to ask. Yes, now second division is meiosis. Specific cells of reproductive organ. As told earlier, this meiosis is limited to only reproductive organ, whether male or female, and it is spatial division for the production of gametes. Okay, gametes are also called spatial sex cells, which are related to the sexual reproduction only, and they are specialized for the fertilization purpose, whether in the plants or animals. Here, specific cells of reproductive organ or tissues in animals and plant divide to form gametes, which after fertilization give rise to offspring. They divide by a different process called meiosis, which involves 2 consecutive divisions. Here, very first difference. You can see mitosis takes place only in 1 cycle of division. But in the case of meiosis, 2 cycles of divisions are involved. Number 1 is meiosis first and number 2 is meiosis second. One more thing before explaining meiosis. 1. let me tell you that meiosis second is totally the copy of mitosis. Exactly the same division. That means meiosis contains two cycles of division. Very first cycle of division is different, but second cycle of division is exactly the division of mitosis. Now, why it contains two cycles? If I'll ask that to whom you resemble the most, or I'll ask, are you the exact or identical copy of either of your parent? You will say no, I resemble both of them in some characters, right? So how these characters mingle together in you? It is just because of meiosis first. Actually, the division, that is meiosis first, contains the mingling. Mingling means the sorting of characters. That means, suppose if I'll show you the diagram, you can easily able to understand. You can see here, very first diagram, it have two kind of chromosome. One is blue in color, one is red in color. But in the very first division, you can see here, they have changed it. That means you can see the part of red one with blue, the part of blue one with the red. That is called mingling. That is mixing of character. Okay. If in case this chromosome is coming in you, it is exactly the same. So you will only resemble one of your parent, either mother or father. But when this chromosome is going in you, that means there is some part of father as well, and other part is from mother. So you will get characters of both. Meiosis have this special step. But one more special point is here. We call mitosis as equational division because it divides into two equal daughter cells. Here, meiosis is called reductional division. Reductional means reduction in chromosome number. If any organism is having chromosome number as 46, then after meiosis, both the cycles, it will become 23 each cell. Let me show you the example. You can see the number of chromosomes in very first cell is 4. Now you can see the daughter cells. Each daughter cell have 2 number of chromosomes. That means just half. And another point, if any chromosome is present in pair, that means 2 set. You can see blue one is present in pair, red one is present in pair. The condition is called diploid. Di means 2, ploid means set of chromosome. And as you know that both have only one set, so the condition is called haploid. Haploid is 1 and ploid means number of chromosome set, that only single chromosome set is present. So this is reductional division. It is detailed diagram for the same. As you can see here, same blue and red color chromosomes are given. In very first division, your mingling take place and both are mixed together. Then the division take place and spindle fiber formation will take place. These spindle fiber attached with the kinetochore pull the chromosomes at opposite pole, and finally your cell divides. Two terms are important. As you can see here, these chromosomes are present in this nuclear membrane, right? Within the nuclear membrane. So it means nucleus is dividing, right? So the division of nucleus is called karyokinesis, as we have studied it in prokaryote and eukaryote as well, that karyon means nucleus. So kinesis means division. Karyokinesis means division of nucleus. So first of all, division of nucleus will take place, then the cytoplasm division will take place. Cytoplasm, that is cytokinesis means division of cytoplasm will take place. So two steps are involved in your cell division, that is karyokinesis and cytokinesis. Here you can see the nucleus is dividing very first, then it is the cytokinesis, your cell is dividing. Here again the mitosis, the similar division, mitosis will take place and all the steps are same. Here you can see both the chromosomes are present, but here no doubling will take place because the step of doubling is done here only. So here as it is cell will come, they will align along the spindle fibers. Spindle fiber will pull them at opposite poles, and finally these four daughter cells. Another division means another point of difference here is four daughter cell. Your mitosis is only producing two daughter cells and your meiosis is producing four daughter cells. Okay, I'll give you one example for the same. If I have 8 toffees with me and I have to divide it equally, so what I'll do, I'll divide it into 4 and 4. Two daughter cells will be produced in very first case, and if the toffees are to be divided into the haploid number, that is 2 by 2 by 2, so I'll give these toffees to 4, then the number will be rich. Here you can see we have initially these 8 arms. 8 means 2 arms, this 2 arms per chromosome. Then it will be divided into these 4 daughter cells. Here is one question from your NCERT book only. Can you think as to why the chromosome number has reduced to half in daughter cells? It is an important case of meiosis. Actually, meiosis is important because it ensures that all the organisms which are produced through sexual reproduction contain the correct number of chromosomes. Why they are just putting more pressure on correct number of chromosomes? Why? Here the correct number of chromosomes is required because each and every organism or species have its spatial number or set of chromosomes. That means if that set is altered by one extra chromosome or one less number of chromosomes than required, it will result in deformities. Okay, in a similar way, if organism producing through sexual reproduction will not give half number of chromosome, suppose let us take an example here, there is one species and it have 2 number of chromosome per head. Okay, so in this condition, if father will give 2 and mother will give 2 as it is, no reduction is there, then it will reach to 4, but that species have its specific number, that is 2, so the number is increased by 2. That means this case will not survive, that kind of offspring will not survive. Let us take our example now. We have 46 as our chromosome number because we have 23 pairs. Okay, so in this case, if both mother and father will give 46, then it will become 92, and this kind of offspring will not at all survive. So that is why we are supposed to do meiosis, produce the gamete and reduce the number to half, so that our species specific number will be reached. That is why meiosis is done. Another significance of meiosis is, as you have seen, that there is mingling and mixing of characters from both parents, that is mother and father. It also results in genetic variation. If there is no genetic variation, we all humans look same, similar, there will be no change. But as we have this meiosis, we have new combinations to be formed during this division, called recombination, that is new combination. Re means again, combinations, that means to combine characters. So by the process of this recombination, we resemble our parent, but we are not identical copy of them. We have our own significance, we have our different characters, because they came from our parents, grandparents, and so on, right? Here is the difference. We have already discussed it during the explanation. Let me revise it. Mitosis occur in all the organism, whether unicellular or multicellular. Meiosis is only restricted to reproductive organs, whether it is plant, fungi, or your animals. Your mitosis helps in division of body cells as it repairs the worn out cell and tissue. On the other hand, meiosis doesn't create any other somatic cells or body cells, only sex cells, which are also called gametes. Mitosis only consists of one cycle of division. On the other hand, meiosis includes two cycles of division. Mitosis do not have any recombination, but meiosis produces new recombinations. Mitosis produces two daughter cells which are equal in chromosome number as the mother cell, but meiosis produces 4 daughter cells which are exactly half the number of chromosomes present in mother cell. Okay, mitosis have all the daughter cells identical, but in meiosis all daughter cells are different. They are different in characters. Okay, here is the cytokinesis which I have explained to you earlier. Here you can see the case of animal cell in which cell division, especially the division of cytoplasm, that is cytokinesis, is taking place here by the growth of plasma membrane towards inwards direction, you can say, and in plant cell the division takes place through cell plate formation. I hope this explanation is sufficient. In case if there is any query or any point which is not understandable, you can ask me. Thank you.